thing will work. Okay. And let's do it. So we're going to start with the conversation about general muscle anatomy, just to make sure that we're all on the same page. So we've got a whole muscle. It's a cross section. Okay. Muscle is surrounded by epimysium. Muscle itself consists of multiple fascicles. And these fascicles, each fascicle is surrounded by the connective tissue called perimysium. And then inside of each fascicle, you have muscle fibers. Each muscle fiber is surrounded by endomysium. So as you can see, there is a lot of similarity between the structure of the muscle and the nerve. They're kind of similar, okay? So these are individual muscle cells, individual muscle fibers, okay? Please do not mix up endomysium and plasma membrane of a muscle cell. Endomysium is all of these are connective tissues. Now, what you can see inside of a muscle cell. So you have, that's the longitudinal section of a muscle cell. So this is the membrane called sarcolemma. Basically, sarco refers to a muscle. Inside of on the sides, you're going to see nuclei. Remember, muscle cells are multinucleated. Okay, inside of the muscle, sort of important structures, you have individual myofibrils. So this is a myofibril. These myofibrils, and terminology can be confusing. So the whole muscle cell is the muscle fiber. These rod-like structures are myofibrils, and each myofibril consists of multiple sarcomeres. In addition to this, we've got plenty of mitochondria. Okay, that's mitochondria. We're talking about skeletal muscle. And a couple more non-envelopes. We have Sarcoplasmic Reticulum. Okay, Sarcoplasmic Reticulum, SR. It stores calcium. And we've got two more kind of important structures, glycosomes that store glycogen, and myoglobin, a protein that stores oxygen. So far, you following me? Good? Okay. Okay, awesome. Now, first, I want to address the basic principle of how skeletal muscle contracts. And for this, we're going to try and recreate, draw a scheme of a sarcomere. That elementary structure. You see, that's kind of important to understand. The myofibril consists of many individual sarcomeres, like a train consists of many cars of a train. So what is a sarcomere? Each sarcomere is bound, so on the sides, there are so-called Z-discs. So sarcomere is between two Z-discs. In the center of the sarcomere the so-called M line. It's all different proteins, and M line holds together thick myosin fibers. Sorry, not myosin fibers, myosin filaments. We're going to name everything properly, color coding. Now, these myosin filaments are attached to Z discs by elastic filaments. Between those elastic filaments are thin filaments here, 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 and here. Okay, so let's name them. So M line, we got it. Z disc, we got it. So these thick red lines are so-called thick filaments made of a protein myosin. These blue lines are thin filaments made of the protein actin. And these orange lines are elastic filaments. Made of the protein titin. Okay. Jake, you don't have to worry about the video. If bandwidth is kind of glitchy, it's okay. Katie covers you. Okay. Can you see the video from me? Jake, can you unmute yourself and say if you can see the video from me? Okay, I presume Jake can see me. I'm recording it anyway. All right, so yes. So here's what happens during the muscle contraction. So look at this. Thick filaments form what is called a cross bridge. Do you see the structures that look like hockey sticks? So these structures basically, these structures are pulling thin filaments towards the M line, bringing Z discs closer to the center. Does that make sense to everybody? Okay. So when Z discs come closer to the center, okay, I have two Jakes. When Z discs come closer to the center, sarcomere shortens. Okay? So usually it is described this way. This part of the sarcomere right here is called a band. And these parts of the sarcomere, well, technically speaking, this is one half of an I band. So you see, this is one half of an I band. This is one half of an I band. So when sarcomere shortens, A band does not go anywhere, stays where it is. But I band becomes smaller and eventually disappear. What's important to understand is that during the sarcomere contraction, during the sarcomere shortening, the size of the filaments does not change. Neither thin, neither thick filaments, they do not change. Elastic, yeah, they do, but these ones, the blue and the red, they do not change. Okay? Now, we mentioned before that skeletal muscle is striated. meaning that it has little stripes if you look at it under the microscope. So these striations is the result of the overlap between the thin and the thick filaments. Okay, and also this A band is much darker because thick filaments, well, they thick and they make A band more, intensely colored. So A band is the dark part of the striation and I bands are the light parts. So one more time, when sarcomere shortens, Z discs come closer to the center and I band disappears. A band stays the same. Length of the filaments does not change. Okay. Now, when all sarcomeres shorten, those myofibrils inside of the muscle cells shorten, and the entire muscle cell shortens. So that, I hope that makes sense. Now we need to figure out, and this is called a sliding filament model, because thin filaments slide relatively to the thick filaments towards the M line. Now we need to figure out how the contraction actually is made possible. What happens during the contraction? To answer this question, we need to discuss two steps in the skeletal muscle contraction. First is excitation of the skeletal muscle, and second is the contraction of a skeletal muscle. So we discussed before that skeletal muscles being stimulated only voluntarily, they are stimulated, they are controlled exclusively by nerves. So here what happens. This here. Where should I start? Here, okay. This is a neuron. Okay, this is the motor neuron. And this is the skeletal muscle. And hold on a second, I made a little mistake here. Okay, that's sarcolemma, the skeletal muscle. Motor neuron releases a neurotransmitter. Here's little vesicles. And it releases neurotransmitter. Here's our neurotransmitter. A neurotransmitter that is released from the motor neuron is always acetylcholine. No exceptions. So when acetylcholine is released from the skeletal muscle, it binds to its receptor, which is essentially a channel. Here's the neurotransmitter bound to its receptor. By the way, if you have any questions, if you want me to repeat something, just say it. Okay? So neurotransmitter binds to its receptor, which is essentially sodium-potassium channels, and neurotransmitter ACH opens these channels. So it's sodium-potassium channel. It opens the channel, and the channel allows sodium to diffuse into the cell and potassium to diffuse out of the cell. There is more positively charged sodium that diffuses in. That essentially leads to the local depolarization. It makes the membrane potential more positive here. This local depolarization is called end plate potential. So far, we good? Now, this whole structure right here, this one, it's called Neuromuscular Junction, NMJ. Neuromuscular Junction. So what happens next? What happens when the end plate potential is formed? If the end plate potential is above the threshold, then it will open voltage-gated sodium channels and sodium will diffuse into the cell. That diffusion will lead to depolarization. Basically, it leads to formation of action potential. So that's sodium in by voltage-gated sodium channels. So that's depolarization. Once this change in the membrane potential reaches its peak, just like in case with the nerve tissue, voltage-gated sodium channels close and voltage-gated potassium channels open, and potassium diffuses out of the cell, repolarizing it. So that's repolarization, and that one here is depolarization. So far, we good? So this depolarization wave is propagated all the way along the sarcolemma until it reaches the structure that I pictured here, kind of pictured here. Okay, this structure right here is called a T-tubule. So that's T-tubule. Basically, T-tubule is an extension of sarcolemma that increases its surface area and allows the action potential to reach within the muscle cell. Turns out that sarcolemma in the T-tubule is really close with the structures of the Sarcoplasmic Reticulum called terminal cisterns. So this is Sarcoplasmic Reticulum here. Sarcoplasmic Reticulum stores calcium. So when action potential reaches T-tubule, through the system of proteins, which you don't need to know here, the system of proteins, okay, it stimulates the opening of calcium channels in the sarcoplasmic reticulum, and calcium diffuses out in the cytoplasm. So let me go over this whole process one more time in a rather slow fashion. So a motor neuron releases acetylcholine at the neuromuscular junction. Acetylcholine binds to its receptor on ligand-gated sodium-potassium channel, opens that sodium-potassium channel, sodium diffuses into the muscle cell and potassium diffuses out. Since there's more sodium that diffuses into the muscle cell, the sodium influx generates local depolarization, which we call end plate potential. This local depolarization, if it goes above the threshold, it starts this basically a positive feedback loop that generating action potential, opening sodium channels, voltage-gated sodium channels, results in depolarization, and then opening voltage-gated potassium channels results in repolarization. The wave of action potential is going across the sarcolemma and it is reaching a T-tubule, an extension of sarcolemma that increases its surface area and allows to get deeper into the muscle cell. When action potential ends up in a T-tubule, the signaling opens voltage-gated calcium channels in the sarcoplasmic reticulum, allowing calcium to leave the sarcoplasmic reticulum and enter sarcoplasm or cytoplasm of the muscle cell. Good? So this is where we stop. This, what we just discussed, this whole process is called excitation. Now, we need to describe what happens in the contraction. To understand what happens during contraction, we need to draw a thick filament first. So this is thick filament, and it consists of a protein myosin. Myosin has two parts. The tail of the myosin forms the filament itself, while the head of the myosin right here has an important enzymatic function. So myosin head has two main features. It can bind to actin and can break down ATP. Now, juxtapositioned to the myosin head is a thin filament made out of actin. Now, I deliberately demonstrated that thin filament, by default, in the relaxed muscle, isn't bound to heads of the thick filament. This is due to the fact that thin filament is surrounded by the protein called tropomyosin. The function of tropomyosin here is to control the binding of actin filament to myosin heads. When tropomyosin is in this position, actin can't bind to myosin. However, tropomyosin has even higher level of control provided by the protein called troponin. Troponin is critical regulatory element of this whole process, since it can bind calcium, that ion that we discussed basically 5 minutes ago when we talked about the excitation process. So let's see what happens when troponin binds calcium. And I will have to rewrite it because I just don't have a space. So troponin binds calcium. So calcium comes into play. When calcium comes into play, this is calcium. It binds to, where's my focal one? it binds to troponin. When calcium is bound to troponin, it initiates a conformational change in the tropomyosin. So now tropomyosin does not block thin filament, and thin filament binds myosin heads. So I'm going to go over this one more time. Normally, well, not normally, when muscle is relaxed, there is no calcium released. Troponin and tropomyosin prevent actin from binding. Well, strictly speaking, tropomyosin prevents actin from binding to the myosin heads. Once calcium arrives into the muscle cell, calcium binds to the troponin. That binding changes the shape of tropomyosin right here, and tropomyosin does not prevent thin filament, the actin filament, from binding to myosin heads anymore. Actin binds to the myosin, and that starts the contraction. So basically, calcium is essential to start the contraction. Does that make sense? Now, what is necessary for the contraction to proceed? So this is called a cross-bridge cycle, and I have a luxury of erasing it, so sorry that you cannot do it on, well, I'll see if I can do it without erasing them. So let's start at the top here. This is the thick filament, and this is thick filament head, just one, okay? Now you see how it is oriented, like it is cocked, like the trigger of a handgun. So this cocking motion is possible because myosin head breaks down ATP. At this point, myosin head broken down ATP into ADP and phosphate. So this is, sorry, I don't like it. Make it slightly bigger. Okay. So here you have ADP and phosphate. Okay. So energy of that breakdown allowed the myosin head to cock back, and now it is binding actin. So, well, what happens next? ADP and phosphate are released from the myosin head. Here's the ADP. Here's phosphate. And here's the myosin head propelling thin filament to the left. Okay? So far, does that make sense? So basically, I don't have anything sufficiently long to demonstrate it well, but think of it this way. If my forearm is the myosin head, it does exactly this, like a molecular model. Now at this point, the movement is over. Now we need to detach the myosin head from the actin. To do this, myosin head has to bind a molecule of ATP. So myosin head binds the ATP molecule. And that binding separates it from the actin. Now, what's the next step? Myosin head breaks down ATP that releases energy, essentially. So it goes from this position to cocked position because it breaks down ATP into ADP and phosphate. That's active. And then the cycle can start again. Does that make sense? So I don't have anything to pull on, so I will try to demonstrate it using just a marker. So imagine that this is the myosin head. So this is the ATP that it's bound. It breaks down the ATP into two pieces, and that provides energy for cocking the myosin head back. It binds to actin, releases ADP and phosphate, pulls it forward. Pulls it forward. In order to separate from actin, it has to bind ATP. It breaks down ATP, cocks back, releases it, starts the cycle once again. So this whole thing is called a cross-bridge cycle. And it is basically one of the interesting features. It is responsible for a condition known as rigor mortis. When dead people, approximately 12 hours after they die, their muscles become very, very stiff because their muscles run out of ATP and actin and myosin cannot separate. Now, basically, it shows you that muscle contraction can occur as long as there are calcium and ATP. Okay, that's it. As long as you have calcium and ATP, muscle contraction can occur. And in order to stop muscle contraction, you need to pump ATP back into, pump calcium back into the sarcoplasmic reticulum. Any questions so far? If not, let's go over the concept of muscle twitch, muscle tetanus, and how we regulate the force of contraction. So, muscle twitch. Twitch is the process. Oh, yeah, I didn't introduce another really important concept. The motor unit. So, muscle twitch is the contraction of a single motor unit. What is a motor unit? Look here. This is a motor neuron. Okay. By the way, I don't know if you are aware, I forgot to mention this. If you, you can watch this conversation in so-called presenter's view. If you move your mouse cursor to the upper right corner, you can have different upper right corner of the screen. You can have a grid view and a presenter view. If you look at the, if you click on the presenter's view, whoever speaks, and that would be me, whoever speaks takes the screen. Does that make sense? Yeah, so you can see how it works. I don't know. Well, we'll figure it out. Okay. So this motor neuron controls several muscle cells. So this, what you see here, is a motor unit. Okay? Motor unit consists of one motor neuron and multiple muscle cells. Motor unit can be small, it can be large. The size of the motor unit is determined by the number and the size of the muscle cells. So what is muscle twitch? It is a contraction of a single motor unit, which consists of three phases. The lag phase, the contraction phase, and the relaxation phase. So lag, contraction, and relaxation. Lag phase is the time when excitation happens. That's the time that it takes for the acetylcholine to be released, for the action potential to be formed, for the action potential to get propagated across the sarcolemma. And that's when the contraction actually starts. Now, muscle twitches and that actually brings us to regulation of the force of contraction. So muscle twitches, I'm going to draw them just in brown. You see in this example, two twitches are separated by the period of relaxation. Now, if the frequency of nerve stimulation increases and the frequency of twitches increases, one twitch can get overlaid by another twitch and by another twitch. And that leads to the summation of twitches. Very much like we observed a temporal summation in the nerve stimulation. So this process, when there is still some relaxation between the twitches, this is called unfused tetanus. And when there is no relaxation between the twitches, and what you see is something like this, this is called fused tetanus. So this means that your muscles cannot, that particular motor unit cannot contract any faster. It generates the maximum force of contraction via the fused tetanus. Obviously, this state of permanent contraction fatigues the muscle really quickly. Okay, so we can regulate the force of contraction by increasing the frequency of twitches, basically through the temporal summation. And how else can we do it? Another way is to increase the number of motor units that are engaged in a particular activity. So this is muscle, and those are individual muscle fibers. I'm not going to draw all of them, but you understand that there's a limited number of muscle fibers in a muscle. The muscle is stimulated. Here's one fiber is activated. It's a really weak contraction. If the signal keeps on coming and other motor neurons are engaged, then more and more muscle fibers, more and more motor units are going to participate in the contraction. So eventually, all muscle fibers in the muscle will be recruited, okay? So this creates maximum tension. You can stimulate the muscle, you can send the nerve signals more and more and more, but it's not going to help, it's not going to make the contraction more forceful, because you simply do not have any more fibers to recruit. Does that make sense? So let's say if your muscle contains 100 fibers and you recruited all 100 of them, that's it. Okay, so it does put a limit on the human physical capacity of, like, the strength, the force that we can develop. Now, before we move on to the third way to increase the force of contraction, I just want to emphasize that small motor units are stimulated first. Where's my razor? So small, then medium, and then large. That's the order of recruitment of muscle fiber, of motor units. Small motor units are engaged first, then medium, then large. Another interesting thing about motor units is that smaller motor units are responsible for forceful activities. For weak activities which are precise. Think of it this way: when you are writing, it's not really a powerful motion, but it's really a precise motion. So this is when you engage small motor units. When you doing a deadlift, this is a powerful motion, and you engage really large motor units. Okay, they can't produce the precision. The motion will be pretty crude, but it's going to be pretty powerful. Now, third way, in addition to increased frequency and increased recruitment, is muscle hypertrophy. So in hypertrophy, you have same number of cells, but cells are bigger. Why pay so much attention to this? When muscles grow, they don't grow because they add new cells. They grow because each individual cell becomes physically bigger. So this is called hypertrophy. The growth in which you increase number of cells is called hyperplasia, and it does not happen in the muscles. So when you see someone like, I don't know, some big bodybuilder, you can kind of think that this guy has basically the same number of muscle cells as you do. His cells are just bigger. Okay, makes sense? Okay, so that's how we regulate the force of contraction. Now, let's talk about different types of contraction from the standpoint of whether or not there is a motion. So muscle contraction can be isometric or isotonic. In the isometric contraction, the tension or the force is less than the load. So for instance, when I'm pushing this wall, wall is not going anywhere. Okay? Am I creating a force? Yes, I am creating a force. But the load that I'm working against is more than the tension that I can create. In the isotonic contraction, tension is higher than the load. So for instance, when I am moving this water bottle like this, the tension that I create is higher than this load. Now there are two types of isotonic contractions: concentric and eccentric. In concentric motion, muscle shortens. In eccentric motion, muscle elongates. Now it may sound relatively odd, but I can explain it fairly easily. Imagine that someone is doing a bench press. When the person pushes a barbell from the chest up, like this, the pectoralis major muscle, the muscle of the chest, shortens. So this, pushing the barbell up, is a concentric motion. When same person eases the barbell back on the chest, pectoralis major, the chest muscle, becomes longer. It was contracted and now it becomes longer. So that's eccentric motion. Since we're talking about muscles, I'm going to throw in some exercise physiology here and there. So it has been established that to increase the muscle volume, the athlete should focus on the eccentric motion. And to increase the force, to increase the strength, without increasing the volume, the athlete should focus on the concentric motion, and in some cases, even avoiding the eccentric. So for a bench press, it's not possible, but for a bench press, it would be pushing the barbell away and then somehow letting it down without any, resistance, okay? For such motion as, let's say, a pull-up, an athlete would have to pull themselves up and then at the top just release the bar. Okay. Are there any questions? Okay. So, okay, we got this. Let's move on to muscle metabolism. I'm going to call it the ATP muscle metabolism is fine. There are essentially three main sources for ATP in the muscle. The first source, the first way to produce ATP, is called direct or substrate level phosphorylation. The reaction is pretty straightforward. You have ADP and you have creatine phosphate. The phosphate is being transferred to ADP, forming ATP, and leaving behind the molecule of creatine. So this process that involves just one reaction is the fastest. way to generate ATP, but it is the least effective because you have basically one ATP molecule generated per molecule of creatine phosphate. Athletes usually run out of creatine phosphate in a matter of seconds. Second, pretty efficient way to generate ATP is through the fermentation, which can be loosely described this way. Glucose is converted to lactic acid and the energy produced in this process generates ATP. So you've got two ATP molecules out of one molecule of glucose. It's the second fastest, more efficient. You see, not the most efficient, it is more efficient. And finally, aerobic respiration. Which can be loosely presented this way. Glucose is oxidized, generating CO2 and water. This reaction, energy that it produces results in the synthesis of 36 ATP molecules, which makes aerobic respiration most efficient, but it's the slowest process. So far, are you with me? Okay. Now, another advantage of aerobic respiration is that it can use lipids or amino acids instead of, hold on, brain fart. Lipids or amino acids instead of glucose. Does that make sense to you? So, fastest, second fastest, slowest. Most efficient, second most efficient, least efficient. Okay? What I wanted to say. Oh yeah, I'm going to throw a couple of words about lactic acid. For many years, people believed that lactic acid contributes to muscle fatigue. Latest research shows no, it does not. Muscle fatigue most likely results from the breakdown of ATP and accumulation of inorganic phosphate. Inorganic phosphate is pretty slow to be removed from the muscle, so that's why we feel fatigued. Lactic acid, basically, if you exercise really heavy, you do accumulate lactic acid, but lactic acid is completely metabolized in about an hour after you completed your exercise. Does that make sense? One more thing to say about this too, I remember. Remember, aerobic respiration requires oxygen. These two do not. Okay? So, if you don't mind, I'm going to compare fast glycolytic and slow oxidative fibers, and then I'm going to ask you for 5 minutes break. I need to check something upstairs, because I'm at home and I'm in the basement. I have no idea what's going out there. So we're going to compare fast glycolytic and slow oxidative fibers. So what is the main way fast glycolytic fibers generate ATP? They mostly use fermentation, while slow oxidative use aerobic respiration. Mostly. So to use fermentation, they can produce ATP pretty fast, but it's not really effective. So they contract fast. While slow oxidative fibers contract slow. And fast glycolytic are engaged in mostly explosive activities. For example, weightlifting. Slow oxidative are involved mostly in the endurance activities, like running. Not even running, I would say jogging. Okay? Which makes fast glycolytic fibers actually strong and large. Slow oxidative are relatively weak and small, and I'm not going to put that yet. These are, okay, these fatigue fast. If you're lifting heavy, you get tired pretty fast. This fatigue slow. Now, some other more sort of molecular differences. Fast glycolytic use a lot of glucose, so they need a lot of glycosomes. Okay? Slow oxidative, on the other hand, use a lot of oxygen, so they need a lot of myoglobin. They also need a lot of capillaries and a lot of mitochondria. Which makes them red, and fast glycolytic are white. I'm going to put at the end because they are not like white, they are whiter. Okay? Does that make sense so far? So, good example of slow oxidative versus fast glycolytic. The muscles of various animals. The most striking examples, if you will look at the color of beef is red, really red, because cows, they walk all day. They basically do a lot of endurance activities. If you look at the alligator meat, it is really, really white, because gator, how does it hunt? By quick attack. It's an explosive motion. Gator cannot chase a prey or something like that. We good so far? Okay. Now, kind of related to muscle metabolism is the idea of post-exercise oxygen consumption. I'm going to talk about it, and then I'm going to run upstairs really quick and return. Post-exercise oxygen consumption. When you exercise, your muscles use a lot of chemicals. They use a lot of oxygen to oxidize glucose. They basically spend oxygen that was bound to myoglobin. They use glycogen. And when you're done exercising, technically the blood flow to the muscle reduces. But muscles keep using oxygen to replenish those chemicals. They use oxygen to restore glycogen storage. They use oxygen to restore myoglobin storage. They use oxygen to burn lactic acid, so basically to replenish those those chemical imbalances. And lastly, when we say muscle operates in anaerobic conditions, like fermentation, that's anaerobic. Okay. It is anaerobic due to one simple physiological feature. When your muscles contract, they squish the blood vessels, and blood simply cannot reach the contracting muscle. The harder they contract, the harder they squish, the harder it is to blood to get to it. So it's kind of paradoxical. When you work out really, really hard, blood doesn't get effectively into the muscles. Okay? Give me 5 minutes. I'll be right back. Okay, a few things that I wanted to say before we move on. So that kind of, main physiological concepts about the muscles. Few terms like sarcopenia, muscle loss. It's normal. It happens in old people. Well, not in you, you're too young, but it definitely happens in me. It starts at about 35. The best way to offset sarcopenia is weightlifting, resistance training, just accumulating the muscle mass. Muscles are always under some kind of muscle tone. They always are slightly contracted. If the muscle loses nerve stimulation, it goes away. It basically is replaced with a connective tissue. So people who have spinal cord injuries, the muscles that are not innervated, that are not stimulated, for instance, someone who's paraplegic and person's legs don't work, muscles in the legs turn into connective tissue, basically into fat. And it's an irreversible process. I talk about a little bit about Duchenne's dystrophy. It's not a lot. I'm not going to include it here because it's kind of, just a general story. What I want to do in the last basically 30 minutes of this improvised class is to talk about smooth muscle. And I want to specifically review the structural differences, the difference in smooth muscle stimulation, and a couple of responses associated with it. So first, structurally, smooth muscle is non-striated cell with one nucleus. It does not have sarcomeres. It's important to understand. It does have thick filaments. And these thick filaments, so that's myosin filaments, they do interact with the thin filaments. So those are thin filaments. It doesn't have sarcomeres, but thin filaments here and here are attached to the dense bodies, essentially the analogs of Z discs. So when smooth muscle contracts, Z bodies come closer. And Z bodies are organized in the muscle in a corkscrew fashion. So each smooth muscle cell, when it contracts, it kind of contracts like you're squeezing the towel. Now, let's address it can be stimulated not necessarily by the nerve tissue, it can be stimulated by nerve signal, it can be stimulated by nerve signal, by chemical signal, hormonal signal, or even mechanical signal. All of it is possible. Now, what I want to discuss in details, and let me think, okay, stimulation, striated, involuntary, I'm just trying to think. Yeah, let's talk about the smooth muscle cell initiation of contraction. So here you have so-called caveolae, that's the cytoplasm of a smooth muscle cell. And this caveolae, they take up calcium. Smooth muscles do not have the very well-developed sarcoplasmic reticulum, so they get calcium mostly from the extracellular fluid. So calcium gets into the cell, and it binds to the protein called calmodulin. Binding of calcium to calmodulin activates it. So here you see activated calmodulin. Okay, activated calmodulin activates another protein, which is called MLCK. So MLCK is myosin light chain kinase. So what's the function of the kinase? Kinase adds a phosphate residue. To what? To myosin light chain. So if this is myosin here, and this is a light chain of myosin, MLCK will facilitate the addition of so it will break down ATP to add a phosphate to the light chain of myosin, and ADP is what's left. Okay, so you see, myosin light chain kinase, when it is activated, adds phosphate to the light chain of myosin. Myosin can now work and bind actin and can contract. Let me go over this process once again. Calcium enters the smooth muscle cell, binds to calmodulin. This binding activates calmodulin. Activated calcium calmodulin complex activates myosin light chain kinase. Myosin light chain kinase being activated phosphorylates, adds phosphate group to light chain of myosin, and that starts the contraction. When you need to stop the contraction, no more calcium. The protein called myosin light chain phosphorylase remove the phosphate, and myosin cannot contract anymore. So again, calcium is necessary, ATP is necessary. Does that make sense? Now, two concepts that I want to explain, not on the board because I can't really draw it well. Yeah, probably I can't. So one concept is how smooth muscle can stay contracted for a long time. Turns out smooth muscle has so-called latch mechanism. So, well, actually, I'm going to just modify some drawings here. When this phosphorylated myosin here binds to actin, okay, and contraction happens. At some point, it loses the phosphate, but turns out when it lost the phosphate, it dissociates with actin really, really slow. And this contracted system of myosin and actin stays contracted for a long time. Okay, basically, it's not so much it stays contracted, the relaxation is really, really, really slow. We're good? Second concept, which one of the most confusing ones, students often get confused, this is why I explain it. It's called stress relaxation response of a smooth muscle. Specifically, imagine a urinary bladder. Okay? it's this size, it gets filled with the urine and it starts to stretch. This stretched, smooth muscle can actually relax, reducing the pressure in the bladder. Does that make sense? It is kind of a strange idea, but imagine this. Imagine that you are inflating a balloon, a rubber balloon, and the tension of the balloon, it increases, and then suddenly this tension goes away because balloon material relaxes. Does that make sense? So this is called stress relaxation response. And when our hollow organs that get extended, when they get extended, they can relax and they can prevent their content from being immediately excreted, expelled, you know. So, for instance, your urinary bladder, when it gets distended, muscle relaxes and you don't feel an urge to go. But at the same time, this relaxed, smooth muscle still can contract when you need to, when you're about to urinate, okay? So it can relax when it is extended and it keeps its capacity to contract when it is extended. We good? And really the last thing to chat about now, something that I just wanted to outline, is the difference between, so smooth muscles structurally, they can be single unit or unitary and multi-unit. So a single unit can be found in the walls of hollow organs. They are connected by gap junctions. Okay? That makes sense? So they are connected by gap junctions and they operate as the functional syncytium. What does that mean? Imagine you have a million smooth muscle cells. They all can communicate with each other via gap junctions. When one cell changes its membrane potential, when one cell starts to contract, all others start to contract as well. So this is why we call it, they operate as a single unit. Okay, now multi-unit muscle cells can be found in the structures like iris or ciliary muscle. They're not acting, well, they're not acting as a functional syncytium. They are much smaller and they are separated from each other. So individual muscle cells are separated, usually not connected by gap junctions. So they would perform much smaller, much more precise activities such as regulating the diameter of your pupil or regulating the diameter of your lens. Does this make sense? That is, quite frankly, that is all for the muscle.